Welcome students. Today we are going to discuss about the anatomy of flowering plants, which is your chapter 6 of your class 11th biology. So according to the NCERT, the syllabus prescribed by NCERT or CBSE, the portion that we will be discussing today is the tissue system in dicots and monocots. So the name of the chapter is Anatomy of Flowering Plants, and the portion which we are going to discuss today is Anatomy of Dicotyledonous root, which is included in the tissue system in dicots and monocots. So here we will see how the various tissues are arranged in a dicot root. So in order to study the tissue systems in a dicot root, what we need to do is we first need to take a transverse section of a root. So as we have studied in a morphology classes, so this is the root. So how we can take a transverse section? So first, with the help of a sharp blade, what we can do, we cut it into various thin slices, and after adding safranin, we can take that slide under the compound microscope, and then we can observe the transverse section and how these tissue systems are arranged in a dicot root. So what are the tissue systems found in a dicot root? So as we have earlier studied that the tissue systems found in a plant is categorized as your epidermal tissue system. The second one is ground tissue system, and the last or the third one is a muscular tissue systems. So in the anatomy of dicotyledonous root, what we will try to do is we will see how this epidermal tissue system, ground tissue system, and vascular tissue system is arranged or are arranged in a dicot root. So in the ground plan of a dicot root, what we can see that these tiny structures or finger-like projections that we can see here, these are the root hairs. So root hairs are present in the outermost layer, that is known as the epidermis. So the epidermal hairs, they are also alternatively called, they are known as the root hairs, and they play important role during the absorption of water and minerals, which we will be discussing during our water absorption chapter of plant physiology. So root hairs, numerous root hairs are present, and the outermost layer of a transverse section of a dicot root is your epidermis. In some books, you might see that it is written as a piliferous layer, or more importantly, another third question, that is the epidermis in root system it is alternatively known as epiblema. It is asked in various entrance examination. So what is the term used to describe the epidermis of a root? So it is a epiblema. Just remember the information. Then beneath the epidermis, we can find this one, this portion. So this portion is known as our cortex. So it is made up of various simple permanent tissues, and what are these tissues of which the cortex is made up of, we will see when we will see the actual cord section of a dicot root. So this is the ground plan or the schematic representation of various tissue systems found in a dicot root. The next one is this circular structure. It is known as endodermis, and these patches, this is your phloem and this is xylem. So this xylem and phloem together constitute the vascular tissue system along with the still, and the ground tissue system consists of cortex. The epidermal tissue system is the epidermis. So this is your ground plan, and if we have prepared a slide and took the slide under the compound microscope and then observe, then what we can see is, so this is the structure. Suppose this is the portion we have enlarged here. So this cord portion we will try to analyze. Just I have mentioned that the outermost layer of the dicot root is epidermis, and it is alternatively known as epiblema. So this epidermis covers the dicot root and numerous unicellular. Just remember, it is important fact that these root hairs are unicellular. So unicellular root hairs are present in the epidermal layer of a dicot root, and beneath epidermis, these large structures or the circular structures, they are known as a cortex, and this cortex is importantly is made up of parenchymatous cells. So try to remember what are the parenchymatous cells that we have discussed in a tissue system. Simple permanent tissue, and parenchymatous cells are distinguished with the presence of your intercellular spaces. So the cortex is made up of parenchymatous cells, and beyond the cortex many layers of parenchymatous cells are present, loosely arranged, intercellular space is present, and the innermost portion or the innermost part of cortex is known as endodermis. So whenever you try to describe all the endodermis, just mention the endodermis is the innermost part of the cortex. So the endodermis, so how we can distinguish between the cortex and the endodermis? Then these endodermal cells are categorized or distinguished with the presence of a Casparian strips. So this is the circular structure that we see here. These are the cells, single cell thickness, and these cells are, they look like just barrel shaped. So they are barrel shaped cells are making the endoderma or the endodermis, and some of the endodermal cells, they become suberized. So what is the meaning of suberized? Suberin gets deposited in the walls, endodermal walls. So these suberized cells of the endodermis are known as Casparian strips. So why they are suberized? Because when we will study about this symplastic and apoplastic pathway of water absorption, then these Casparian strips do not allow the water to penetrate beyond the endodermal layers. So, but we know that the vascular bundles are present beyond endodermis. Then how these waters are get transported by the xylem, that we will be discussing in some other chapters. So they make impermeable, they are the water impermeable membranes or the structures found in the endodermis. Then next to endodermis, we will see the presence of pericycle. So pericycle is more than one layer thickness. And here you have to remember the function of pericycle. So what does pericycle do? So this pericycle, the layers of the pericycle, they give rise to lateral roots. So this is the primary root. As it is a dicot root, so we will see the presence of secondary growth in dicots. So these pericycles, they give rise to lateral roots, and these lateral roots along with the vascular cambium makes the secondary growth in dicot roots. But when we will study about the monocot root, then the pericycle will give rise only to lateral roots but not to your vascular cambium. So that's why in monocot roots we will see that there is no secondary growth. Then next we will come to your vascular tissue system or the vascular bundles. So the vascular bundle is made up of the xylem and phloem. So the xylem and phloem make the vascular bundle, and usually these vascular bundles, the xylem and phloem, they are found in patches, generally 2 to 4 patches. So accordingly these are variously torn, like your, if the vascular bundles or patches are up 2, then it is diarch. If it is 3, then it is triarch, 4 tetrarch. And in monocot roots we will see that these vascular bundles are of polyarch nature. What is polyarch? It is the number of patches is greater than 6. So more than 6 number of patches are found in monocot roots. But here in dicots it is generally 2 to 4 patches. Then the arrangement of xylem we will see, it is of your exarch condition. Exarch or exarch condition. So what does this term explain? This term explains that the first form cells or the protoxylems are found towards the epidermis or towards the periphery. So these are the xylems. So this is called a metaxylem. This is a protoxylem. So the protoxylems are present towards the center, towards the periphery. If you observe this structure, so this is metaxylem and these are the protoxylem. So the protoxylems are present towards the, or found towards, close to the endodermis or epidermis, towards the periphery, and the metaxylem is found towards the fifth. So this type of arrangement in which the protoxylems are found towards the periphery or epidermal region and metaxylem towards the central region, this type of arrangement is an exarch condition. So here the protoxylem is of exarch condition. Then in between the xylem and phloem, so this is the xylem and here is the phloem patches. So 1, 2, 3, 4 patches, xylems are showing 4 patches. So in between the xylem and phloem, so these are phloems, this xylem. What we will see? These structures. What are these? These are the parenchymatous cells. So these parenchymatous cells present in between the xylem and phloem. Then next we will come pith. As we will see here, very small or inconspicuous pith is present. So pith is very small in dicot root. So the next point is your pericycle, then your vascular bundle and pith. So this pith, pericycle and vascular bundle, they together known as your stele. What is it? It is stele. So they constitute stele. So this is all about the arrangement of various types of tissues in a dicot root. So next we will try to summarize the concept that we have studied just now. So in order to describe the anatomy of dicotyledonous root, we have to prepare a slide or prepare a chord section, that is transverse section. So how we will go about describing the structures? First, we will try to describe each tissue from your epidermis to your pith. So from epidermis, what we will see? Epidermis contains some root hairs. Then next part is a cortex. Then endodermis, pericycle, muscular tissue. Then about a pith and how the muscular tissues are arranged here. So in a dicot root, we will see the epidermis is the outermost layer, and the epidermis contains numerous single cell or unicellular root hairs. Cortex is of parenchymatous and containing intracellular spaces. And next to cortex is a single layer thickness endodermis, and this endodermal cells gets suberized, and these cells are known as your Casparian strips. They don't allow water to penetrate. And next is your pericycle. The pericycle in dicot root give rise to your lateral roots along with the muscular cambium. That's why it plays important role in the secondary growth. And next is your vascular bundle, usually found in 2 to 4 patches according to the various terms, diarch, triarch, tetrarch, hexarch, and in monocots it is of polyarch nature. And in between the xylem and phloem we will find the conjunctive tissue. So they are known as a conjunctive tissues. They are made up of your parenchymatous cells, and pith is very small or inconspicuous, and pericycle, vascular bundle along with the pith is known as our still. So what questions we will expect from this anatomy of dicotyledonous root? So first one is, if we will try to understand how we can approach the question. So first one is, if the question is about explain the anatomical features of a dicot root. So here, as just before I was giving the summary of the anatomy of dicotyledonous root, just go on writing starting from epidermis to the pith. Then next one is from this portion we can expect questions about the what is exarch, what is diarch, short questions like your root hairs are of unicellular nature. Then cortex is of the parenchymatous nature. Then the xylem and phloem are arranged in various radii. So what is the term used for that? So here we can see the last question is the importance of Casparian strips. So just go through your textbook and practice drawing these diagrams. It's very easy, and with practice we will make these diagrams under a very short span of time. I hope this gives you clarity about the various tissues arranged in a dicot root, and thank you all for listening this or watching this video. In the next class we will try to understand the anatomy of monocot root and how these monocot and dicot roots differ in their structure and along with their function. Thank you.